Okay, so nucleic acid, I have told you nucleic acid is a biopolymer. Okay, polymer means it's a large chain molecule containing many small units. Okay, nucleic acid, it is also called as polynucleotide. Okay, where nucleotide is the monomer, right? Many nucleotide unit joined together and form what is called a large molecule of nucleic acid. So basically nucleic acid is a polymer, right? Then in nucleic acid, what nucleic acid will do? Nucleic acid will store the information. Nucleic acid will also transfer the information. It stores and transfers the genetic information. Okay. Students, is there any lagging? No. Okay. Right. So nucleic acid is a very important one for human cell and plant cell. What they will do, they will store the genetic information, they also transfer. Then eukaryotic cell, you know very well, higher organisms have cell called eukaryotic cell. Eukaryotic cell has nucleus, whereas lower organisms, amoeba, certain viruses, they have what is called prokaryotic cell. In prokaryotic cell there is no nucleus. Okay, in eukaryotic cell we have nucleus. All nucleic acids, Rna and Dna, very specifically Dna, right, are present inside the nucleus. As nucleic acids are present as chromatin. What is chromatin? Chromatin is nothing but nucleic acid plus proteins. Okay, nucleic acid along with some protein is what we call as chromatin. Okay, this chromatin, right, is actually called as chromosomes. These chromosomes are responsible for the genetic information. Okay, then what we are going to do in chemistry is that we are going to discuss about the chemical composition of nucleic acid. Right? See, when you suppose you take some nucleic acid, it may be RNA or maybe DNA. We are going to discuss that. If you hydrolyze completely, if you do complete hydrolysis of nucleic acid, we will get sugar plus base plus phosphoric acid. Phosphoric acid has on hydrolysis we will get phosphoric acid. Okay, so keep this in mind. Nucleic acid is a polymer. If you hydrolyze this polymer completely, you will get many molecules of sugars, many molecules of bases, and many molecules of phosphoric acid. So what does this indicate? It indicates that sugar, base, and phosphoric acids are the building blocks of nucleic acids. Okay, now, and in nucleic acids, I have told you there are two types. One is RNA, the other one is DNA. What is RNA? RNA is nothing but ribonucleic acid, and DNA is deoxyribonucleic acid. Okay, then in RNA as well as DNA, I have told you, if you hydrolyze completely, you will get sugar, you will get bases, nitrogenous bases, then you will get phosphoric acid. Okay, first we will discuss about sugar. Okay, what type of sugar is present in nucleic acid, and what is the difference in sugar which is present in RNA and DNA. Okay, keep this in mind. Sugar present in RNA, sugar present in DNA are not exactly same. Okay, there is some difference. What is that? We will discuss. Sugar, the sugar that is present in nucleic acid is actually the pentose sugar. Pentose means it contains 5 carbon atoms, right? And it exists in cyclic form called furanose form. Okay, ribofuranose. The full name is ribofuranose. Why I am using the word ribo? Because the sugar, the pentose sugar that is present in nucleic acid is actually ribose. Ribose. It is actually ribose. Okay. Now, in RNA, we have the ribose sugar. Ribose sugar is nothing but aldopentose. Okay, and this is the open chain structure of ribose. Open chain structure. See the structure, it has totally 5 carbon atoms. Here I have given the Fischer projection of ribose. In Fischer projection, the first carbon is actually a part of aldehyde group. Okay, and 3 carbon atoms are chiral carbons, and last one is a primary alcoholic group. This is not a chiral carbon. Okay, students, turn on your camera, everyone. Turn on your camera. Yes. Right. So this is called D-ribose, where OH groups are present in the right hand side in Fischer projection. Now, in cyclic form, when it forms cyclic structure, how it forms cyclic structure? How does? Listen, this OH group of carbon number 4 will react with the aldehyde group. Students, observe this carefully. Reaction between aldehyde and alcohol, okay, will give you a compound. That compound is called hemiacetal. Hemiacetal. But here, aldehyde group and alcohol group are present in the same carbon, same molecule. I'm sorry. Aldehyde group and the OH group are present in the same molecule. So this gives you what is called cyclic hemiacetal. Right? So when this OH group, there is OH group of 4th carbon. Okay, if it attacks this aldehyde group, right, this aldehyde carbon become a chiral carbon. Okay, that carbon is number 1. In sugar, students, this is very important. In nucleic acid chemistry, we give 1 prime, 2 prime, 3 prime, 4 prime, 5 prime numbering for sugar. For bases, we give 1, 2, 3, 4 normal numbers. Okay, here you should use primes numbers. Okay, and this carbon number 1 prime carbon, which is originally aldehyde carbon, but now due to the formation of cyclic hemiacetal structure, that 1 prime carbon become an anomeric carbon. Okay, so you'll get, due to the cyclic ring formation, you'll get an OH group here. That OH group may present above the ring or below the ring. If it is present above the ring, we call it as beta. Okay, then you know very well, whatever OH group present in the right hand side, that should be kept below the ring. Okay, so in second carbon, OH group is in right hand side, so place it down. In third carbon also, OH group is right hand side. Okay, place it down. Right, and in fourth carbon, we have one hydrogen, one CH2OH group. Always place hydrogen down and CH2OH above. Okay, so this is the ribose sugar actually present in RNA. Students, keep this in mind. In RNA, right, the ribose sugar is actually present in cyclic form, and it is called, this cyclic form is called beta D ribose. Okay, and remember the structure. In carbon number 1, OH group is above the ring. Carbon number 2, OH group below. Carbon number 3, OH group below. Carbon number 4, hydrogen below, CH2OH above. That CH2 carbon is actually 5 prime carbon. So it is a pentose sugar. It contains 5 carbon atoms. See the ring? The ring is 5-membered ring. Okay, in ring we have one oxygen atom here. So remember the structure, which is very, very, very important. Right? Then, so this is the RNA. What about DNA? What is the difference between the sugar present in RNA and sugar present in DNA? Listen, in DNA also we have the same sugar. In DNA also we have the same sugar with a small difference. What is the difference? The difference is that in carbon number 2, listen, I kept the ribose structure as such. This is also beta D ribose only. But what is the difference? The difference is, from the second carbon we have removed one oxygen. Okay, from the second carbon we have removed one oxygen. Therefore it is called beta D 2-deoxy. 2-deoxy means oxygen is removed in second carbon. Beta D 2-deoxyribose. This is the sugar present in DNA. So what is the difference in sugar present in RNA and DNA? In RNA we have normal ribose, beta D ribose. In DNA we have beta D 2-deoxyribose. Okay, and this is regarding sugars. Next one, bases. What are the bases present in nucleic acids? Okay, actually we have two family of bases. The bases present in nucleic acids are nitrogenous bases. Nitrogen containing bases. Simple. Nitrogenous means nitrogen containing bases. In nucleic acid we have two family of bases. One is purine, the other is pyrimidine. Okay, this is the basic structure of purine. I have told you purine is a bicyclic compound. See, this is one cyclic ring, this is another cyclic ring. One is 6-membered ring, another one is 5-membered ring. So it is a bicyclic compound. Pyrimidine is a bicyclic compound. I am sorry, purine. Purine is a bicyclic compound. Whereas the other one is pyrimidine. Pyrimidine is monocyclic. Pyrimidine is monocyclic. Okay, and see the numbering here. Number starts from this nitrogen 1, 2, 3, 4, 5, 6, then 7, 8, 9. We have totally 9 atoms in the ring. We have totally 9 atoms in the ring in purine. In pyrimidine we have only 6 atoms. I am giving a number in this in anti-clockwise direction. 1, 2, 3, 4, 5, 6. Okay, and see, in purine we have 4 nitrogen atoms. Right? How many nitrogen atoms are there? In purine we have 4 nitrogen atoms. 4 nitrogen atoms in purine. Okay, where they are present? Actually, they are present in odd number position. Odd number position 1, 3, 5 is fused between the two rings, so nitrogen cannot present there. Therefore, what are the positions? The positions are 1, 3, instead of 5, okay, it present in 7 and 9. 5 position is in between 6-membered ring and 5-membered ring. So, in that position, nitrogen is difficult to present with this. Therefore, or just keep this in mind like this. The nitrogen atoms are present at 1, 3, 7, 9 positions. Okay, in purine bases. In pyrimidine bases, nitrogen is present at position number 1 and 3. Okay, here we have 2 nitrogen. We have 2 nitrogen, and the nitrogens are present in 1, 3 position. Here 1, 3, 7, 9 position. Remember the structure. Remember the position of nitrogen atoms. Now, in this 2 family, what are all the bases actually present? Okay, we have 2 family of bases, purine bases and pyrimidine bases. First, let's start with the purine base. Okay, purine bases. I have kept the basic structure as such. What I did here, I have just introduced one amino group at position number 6. I have introduced one amino group at position number 6. So this compound is called 6-aminopurine. 6-aminopurine. 6-aminopurine is nothing but adenine. Okay, this purine base is called adenine. We use the symbol A for adenine. And second base, what I did here is I placed NH2 group at position number 2 now. And in position number 6, I put an oxo group, keto group. See, double bond O means keto. Whenever keto group is present, students, keep this in mind. The adjacent nitrogen should contain one hydrogen atom. And there should not be any double bond between these two. Whenever keto group is present, adjacent nitrogen should contain one hydrogen. Right? Keep this information. This is very useful for nucleic acid. And this compound is called 2-amino. In second carbon we have amino group, so 2-amino. 6th carbon we have an oxygen, so 6-oxopurine. 2-amino 6-oxopurine is called guanine. It's called guanine. And we use the symbol G for guanine. Okay. So in purine base, in purine we have two bases. What are they? Adenine and the guanine. Right? Then what about pyrimidine bases? In pyrimidine we have three bases: cytosine, uracil and thymine. Students, here this is a very essential one. Often students may write like this. Instead of thymine, they will write thiamine. What is thiamine, students? What is thiamine? Thiamine is the base present in nucleic acid. Thiamine is actually vitamin B1. Okay, so keep this in mind. You should not write thiamine. This is thymine, T-H-Y. I'm very sorry. It's not thymine. This is thymine. I like to write here. This is thymine. Thymine is the base present in nucleic acid. Okay, often by mistake students will commit this one. Okay, what they will do, instead of writing thymine, they will write thiamine. Thiamine is vitamin B1. It is not the base present in nucleic acid. Okay, so keep this in mind. And remember this shortcut, CUT, cut. CUT, cut. Cytosine, uracil, thymine. Okay, now listen, I'll just show you how to draw these structures, pyrimidine bases, just for understanding purpose. Students, just observe this carefully. How to draw the structure of pyrimidine bases. Okay, the shortcut is this, CUT, cut. C means cytosine, U means uracil, T means thymine. These are the three bases present in nucleic acid. Okay. Now, all these three pyrimidine bases, these are all the pyrimidine bases. All these three are pyrimidine bases. Okay. In these pyrimidine bases, okay, what is the common structure? Common structure is this. See, students, observe carefully. Nitrogen, nitrogen. Okay, 6-membered ring. Similarly here, nitrogen, nitrogen, 6-membered ring. Here also nitrogen, nitrogen and 6-membered ring. This is actually common. And put a double bond here. Double bond, double bond, double bond. In this position, okay? And put a keto group here. C double bond O, C double bond O, C double bond O. These are all common. And here one hydrogen, here one hydrogen, here one hydrogen. Okay, see, up to this there is no difference. There is no difference. All these structures have common entities. Okay, now I am going to make some substitution. In cytosine, see, this is number 1, this is number 2, this is number 3, this is number 4, this is number 5 and this is number 6. Okay, you just give the number in anti-clockwise direction. In 4th position, I'll put an amino group. In 4th position, I'll put an amino group. What is this? This is called 4-amino pyrimidine. I'm sorry, 4-amino 2-oxo. 4-amino 2-oxo pyrimidine. 4-amino 2-oxo pyrimidine. This is what we call the cytosine. For uracil, what you have to do? Remove the amino group and put a keto group here. I have told you, whenever you put a keto group, the adjacent nitrogen should contain nitrogen. Nitrogen should contain nitrogen. And this is the structure called uracil. This is the structure of uracil. In uracil, see, this is 1, this is 2, this is 3, this is 4. Okay. In 2nd carbon we have an oxygen and 4th carbon we have an oxygen. So it is called 2,4 dioxo. We have oxygen atoms at 2 positions. Okay. So 2,4 dioxopyrimidine. This is called uracil. 2,4 dioxopyrimidine is called uracil. And what about thymine? In thymine, as the basic structure of uracil, it looks exactly like uracil, And in fifth position, this is fifth carbon, we should put a methyl group here, CH3 methyl. So what's the name? The name is this 2,4 dioxo 5-methyl pyrimidine. It is what we call as thymine. Let me write here 2,4 dioxo. Okay, 5. I don't have space here, so let me write here 5-methyl. Okay, pyrimidine. Yes, 5-methyl pyrimidine, and this is what we call as thymine. Okay, so students, just remember the common structure first, then you place the substituents so that you can easily remember the structures. Okay, so now you know what are all the pyrimidine bases present, right? Then in the last class I have told you not all these bases are present. See what I am trying to say? We have purine bases, we have pyrimidine bases. How many purine bases are there, students? Can you tell me how many purine bases present in nucleic acid? Purine. Yeah, 2 purine bases. What are they? Adenine and guanine. How many pyrimidine bases? Yes, answer is here. Yeah, we have 3, right? Cytosine, uracil, thymine. So totally 5 bases are there. But among the 5, only 4 are present in nucleic acids. Okay? What are the 4? What are the 4 present in nucleic acids? So bases, we have totally 5 bases: adenine, guanine, cytosine, uracil and thymine. Adenine and guanine are purine bases. Cytosine, uracil and thymine, they are pyrimidine bases. Okay, so totally we have 5. Among the 5, only 4 are present in RNA and 4 are present in DNA. What are the 4 bases present in DNA? See, adenine, guanine, thymine and cytosine. Okay, in RNA, except the thymine, all other bases are same. Adenine, guanine, cytosine. Instead of thymine, we have uracil in RNA. Okay, so keep this in mind, very, very important. In DNA, we have AGTC, whereas in RNA, we have AGUC. Okay, yeah. So keep all these things in mind, very, very important. And what is the basic difference between RNA and DNA? So far, we discussed sugar and bases. Based on these two, I am going to say RNA is single strand. It contains single polynucleotide chain. RNA contains single polynucleotide chain, whereas DNA is a double-stranded. Okay, 2 chains. Okay, they wound around each other, right? And exist like this. This is called double-stranded. Double-stranded means 2 polynucleotide chains. They wound around each other. Okay. Then in RNA we have beta D ribose sugar. In DNA we have beta D 2-deoxyribose sugar. Then in second important point, in RNA we have adenine, guanine, uracil and thymine. We have uracil here. In DNA, instead of uracil we have thymine. That's all. So AGTC. Right? These are the basic differences between RNA and DNA based on the chemical composition. Okay, right. Students, so far any doubt? Yeah, right. So now what I am going to do, next important point is this. So I have told you about sugar that is present in nucleic acid and bases present in nucleic acid, and the last one is phosphoric acid. Okay, this is the structure of phosphoric acid, right? Students, keep this in mind. In nucleic acid, we don't have actually phosphoric acid. We have phosphate. So phosphoric acid exists as phosphate in nucleic acid. That's the point. But if you do hydrolysis, you'll get phosphoric acid only. Now, nucleic acid, I have told you, it's nothing but polynucleotide. And the nucleotide is the monomer. Is the monomer unit. Many nucleotide join together and you will get what is called polynucleotide. Okay. What is nucleotide? What is mean by one nucleotide? One nucleotide means it contains one sugar molecule, one base molecule and one phosphoric acid. If these three combine, you will get what is called nucleotide. If you combine one sugar and one base without phosphoric acid, we call this unit, this combined unit is called nucleoside. Students, remember the name nucleoside, nucleotide. Nucleoside means sugar base only. The combination of sugar and base. Whereas here the combination of sugar, base and phosphoric acid. Okay. Now, right, where this base will attach and the phosphoric acid will attach in the sugar unit. Okay. So I have taken RNA as an example, and you know very well in RNA we have ribose sugar. Right? Students, keep this in mind. Always base molecule will join with the 1' carbon of sugar. Okay, and phosphoric acid unit attached with the 5' carbon of sugar. This is 5' carbon. Okay, I repeat. Base will join with the 1' carbon of sugar, and phosphoric acid will connect with the 5' carbon. You will get what is called nucleotide. So that's why I wrote here C1' carbon of sugar is connected with the N9 position. Okay, 9th position nitrogen is connected with the 9th position nitrogen in purine and our first position of nitrogen in pyrimidine. Okay, what I am trying to say? Listen. Let me just draw one nucleoside and one nucleotide. Okay, first I'll take a nucleoside. Students, observe carefully. Nucleoside. Right? Let us take a nucleoside in RNA. If I say RNA, the point that immediately comes to your mind is the sugar present in RNA is ribose sugar. And what are the bases present? Sugar is ribose. What are the bases present? Adenine, guanine, instead of thymine we have uracil and cytosine. AGUC. These are all the bases present. Okay. Now I'll take adenine base just for understanding purpose, and I'll show you how to draw a nucleoside. Okay. Students, observe carefully. Nucleoside contain a 5-membered ring of sugar, where this is 1' carbon, 2', 3', 4' carbon. In 1' carbon, we have OH group above the ring. That's why it is called beta. Then we have OH group below in the second carbon, OH group below in third carbon. In fourth carbon, hydrogen below, CH2OH above. This is ribose sugar. Okay. And what is nucleoside? I have told you. Nucleoside is nothing but a combination of sugar and base. When a sugar and a base joins, you will get what is called nucleoside. Now I have taken sugar and now I will take base. I will draw the base here. So we have, I have taken adenine as an example. So let me draw the structure of adenine. Adenine is a bicyclic compound. Okay, and here in adenine we have double bonds. Let me put the double bonds here. Double bond. Double bond, here one double bond, and in this position we should put an amino group. Okay, 6 amino purine. This is called adenine, right? And here we have double bond, and here we have nitrogen. This is the nitrogen present in 9th position, and this is the OH group present in 1' carbon. Now listen, this hydrogen and this OH group, they react and eliminate a water molecule. Okay, so H and OH will be removed and you will get a new bond between this nitrogen and this carbon, and that unit is what we call as a nucleoside. Okay, let me draw that. See how the structures will look like. Students, observe carefully. Right? I'll keep all other carbon as such. CH2OH, H, OH, OH. I'll keep all the carbon as such. Here, this hydrogen I'll keep it as such. I'm going to remove this OH and hydrogen, and I'm going to connect this to the nitrogen. Okay, so let me draw like this. Here we have a 6-membered ring, and here we have a 5-membered ring. Nitrogen. Okay, let me keep all this as such. This is adenine structure. Now adenine is joined to the sugar unit. Okay, this is 1' carbon, and this is 9th position of nitrogen. Okay, you will get what is called a nucleoside, and this nucleoside is called adenosine. We have adenine, so it is called adenosine. Okay, adenosine. This nucleoside is called adenosine. Now I will write the structure of a nucleotide. Nucleotide. What is nucleotide? Nucleotide contains 3 units. What are 3 units? Sugar plus base plus phosphoric acid. Okay, plus phosphoric acid. Phosphoric acid. Okay, so what I'm going to do, I will take this nucleoside, and in 5' position, this is 5' position, this is 1', 2', 3', 4', and this is 5' position. In 5' position, with this OH group, I am going to join a phosphoric acid unit. So what will happen? See here. Students, let me write once. This is phosphoric acid, P double bond O, OH. First I will write phosphoric acid, OH, OH. Okay, this is phosphoric acid. Now I am going to connect this here. Okay, this is OH group of CH2, that is 5' carbon. Now it is connected to the 5-membered ring. Let me draw like this. Okay, and here we have the base. What's the base present? The base is actually adenine now. Okay, I have taken this example, so this base is now adenine. Right? Keep all other things as such. Keep all other things as such. Now, students, observe carefully. Now what I am going to do, I am going to listen. This OH group react with this OH group, and they eliminate a water molecule. So remove one water molecule here, H2O, and we have an oxygen. That oxygen connect the phosphorus with the CH2. Okay, and remember, inside the cell, okay, we have slightly basic medium. Therefore, the OH group never present as acid, they actually present as O- ion as a salt. So, as phosphate. Okay, so let me draw the structure here. We have P double bond O. Instead of OH, we'll put O minus. And here we have a new connection between this phosphorus and this CH2 group. Okay, and that is connected to the 5-membered ring like this. Okay, and here we have the base unit. Students, observe carefully. This is 5' unit. Okay. Now in 5' unit we have one phosphate group. Okay. If the base is adenine, suppose if the base is adenine, then this is called adenosine monophosphate. Students, turn on your camera. Are you able to follow this? Suppose if the base is adenine, turn on your camera. Suppose if the base is adenine, then it is called adenosine monophosphate, AMP. Okay, or I can put even 3 phosphate unit here. If I put 3 phosphate, if I join 3 phosphate, it is called ATP, adenosine triphosphate. Okay, if I take, suppose instead of adenine, if I take guanine, I will get guanosine monophosphate. Okay, so this is what we call as a nucleotide. So let me just redraw in a simple manner how a nucleotide will look like. A nucleotide has the structure like this. It has a sugar unit. In 1' carbon, a base is attached. Okay, this is 1' carbon, right? And in 5' carbon, this is 5' carbon. In 5' carbon, a phosphoric acid unit is attached, or phosphate unit. PO, O minus, right? The phosphate unit is attached in 5' carbon. This is 5' carbon. This is the basic structure of nucleotide. This is one nucleotide. If I join many nucleotide, okay, we will get what is called a polynucleotide. Okay, how a polynucleotide is formed? Listen, students, observe carefully. The OH group of third position, that is 3' carbon. In 3' carbon, we have a OH group, right? We have OH group. Now what will happen? Another molecule of nucleotide, let me show here. Students, observe this carefully. I'll take another molecule of nucleotide. We have a base, right? Here we have carbon CH2, P double bond O minus. Okay, this is another molecule. This is one nucleotide. This is another nucleotide. Now what will happen, students? One nucleotide and another nucleotide, they join. How? This O minus will attack this carbon so that this OH group will be removed. You will get a new bond or new connection between third carbon, 3' carbon of ribose with this oxygen. So you will get a structure like this. See, students, observe the structure carefully. It's a pentose sugar. Here we have a base unit. Okay, second carbon. In third carbon, now we have hydrogen above. Now this OH group is removed and you will get a new connection with oxygen. And here we have P double bond O and O minus is there, and that P is connected to CH2 of this ribose, and that CH2 is connected to this pentose sugar. Okay, students, is this clear? Any doubt? This is 5' carbon of second ribose unit, and here we have CH2 P double bond O minus. This is the first nucleotide. This is the second nucleotide. They join together through a phosphate unit. Okay, between one nucleotide and another nucleotide, what is the connection? 3' carbon of one ribose, 3' carbon of ribose in one nucleotide is connected with the 5' carbon of ribose in another nucleotide through a phosphate. Students, observe this carefully. P in organic chemistry, C double bond O. This group is called ester group. Okay, P double bond O, single bond O. This is called phosphoester. Okay, phosphoester. Suppose if I have one more oxygen this side, we call this as phosphodiester. Phosphodiester. Okay, so here see, I have P double bond O. This side I have an oxygen. Okay, yeah, I'm very sorry, I made a mistake. Okay, so between phosphorus and this CH2 there should be one oxygen, right? Similarly here, between phosphorus and CH2 one oxygen should be there. I missed it. Let me put that. We have an oxygen here. So between phosphorus and this CH2 there should be an oxygen. Okay, so here we have P double bond O. This side also we have P double bond O. So it is a phosphodiester is present between these two nucleotides. So the point is this: the two nucleotides are connected through a phosphodiester linkage. Okay, this is called phosphodiester linkage. Students, observe carefully. In nucleic acid, one nucleotide and the adjacent nucleotide are joined together through phosphodiester linkage. Very, very important. Okay, right? Then the OH group of this carbon, this ribose that connected to the next nucleotide, so the chain goes like this. Similarly, here also the chain goes like this. You will get what is called the polynucleotide chain, also called nucleic acid. Is this clear, student? Any doubt? You require some practice in this, so you just write and practice afterward. Do you have any doubt up to this, students? Any doubt up to this? Okay. Now listen, students. When you move from one nucleotide to another nucleotide, okay, if you move along this chain, see, phosphate, sugar, these are all common. What is the difference? In a given, okay, in a given nucleic acid, suppose you take RNA. In RNA, okay, this phosphate, this sugar, this phosphate, sugar, these are all common features. What is the difference? The base. Base alone will differ. Okay, base alone will differ. So, if I write a polynucleotide chain like this, just for understanding purpose, students, look carefully. Suppose I'll take RNA. In RNA, I'll write a polynucleotide chain like this, right? And first nucleotide contains some sugar, I'm sorry, some base, maybe adenine. Second may contain guanine. Third one may contain cytosine. Fourth one, since it is RNA, uracil must present uracil. Okay, like that it goes, adenine, like that. It need not be present in the same order. Okay, it can be of any order, but it actually we represent like this normally. Okay, this is a polynucleotide chain. A polynucleotide chain. Listen, student, this is very, very important. Students, all of you, please turn on your camera. Turn on your camera. This is very, very important. In RNA, we have only single chain, whereas in DNA, we have two chains. We have two chains. Okay. In one chain, suppose I have adenine, here I have guanine, here I have cytosine, thymine. Okay. Similarly, here I have adenine, thymine. It need not be the same order. I told you, it should be different. But here, which is very, very important, one terminal nucleotide of one chain, okay, you take one chain, the one terminal nucleotide has phosphate unit free at 5' carbon. 5' carbon phosphate unit present as free phosphate. And the other end, we have 3' OH group that is present free. Correct? When we move from one nucleotide to another nucleotide, the 3' OH group will react with the next nucleotide, so that the chain continues. At the terminal position, okay, the nucleotide present at the terminal position, it has free OH group. That OH group remains as such. So normally we represent polynucleotide chain like this: 5' at one end, 3' at another end. What is the meaning? The meaning is 5' means this nucleotide, that is the terminal nucleotide, has free phosphate group at 5' position. This terminal nucleotide has free OH group at 3' position. So here we have free OH group. Here we have free phosphate unit. Okay, and students, observe this carefully. Always we see nucleic acid from this direction, from 5' to 3'. Okay, and in DNA, this is a very important feature. In DNA, we have two polynucleotide chain. If one chain goes like this from 5' to 3', the second chain will go like this. Okay, that is 5 prime to 3 prime. It is going in the opposite direction. This is called anti-parallel. In DNA, the two nucleotide chains are anti-parallel. Students, observe carefully. They are anti-parallel. We have two strands of polynucleotide chains, but they exist in anti-parallel manner. But why it is called anti-parallel? Because we by convention we see nucleic acid from 5 prime end to 3 prime end. So 5 prime to 3 prime, this is one direction. In the other nucleotide chain, the end will go like this. It starts from the 5 prime end will be in the opposite side, and 3 prime end will be the other side. It is exactly opposite to this chain. That's why in DNA the two nucleotide chains, okay, are anti-parallel. And one more information, student, observe carefully. Always the two chains are connected. How they are connected? They are connected through hydrogen bonding. Okay, two chains in DNA, the two nucleotide chains are connected through hydrogen bonding. How? See, the base present in the other nucleotide chain, right? Always with the base adenine in one nucleotide chain, the base thymine will join. Students, observe carefully. Between one nucleotide chain, one polynucleotide chain and another polynucleotide chain, okay, how these two are connected? They are connected through hydrogen bond. How? The hydrogen bonds are formed between the bases. Okay, adenine of one polynucleotide chain will form hydrogen bond with the thymine of another polynucleotide chain. Okay, similarly, guanine of one polynucleotide chain will form hydrogen bond with the cytosine of another polynucleotide chain, and there is a hydrogen bonding between these two. What type of hydrogen bonding? See here, it is actually intermolecular hydrogen bonding, no doubt in that. Between A and T, we always have two hydrogen bonds. Two hydrogen bonds, okay, get formed between adenine and thymine always. Between guanine and cytosine, we get 3 hydrogen bonds. Okay, I didn't draw the full structure here also. For cytosine always combine with guanine. Thymine always combine with adenine. Adenine, thymine. Thymine, adenine. Guanine, cytosine. The pair will be like this always. Okay, just remember this. ATGC. Always the pair is this. Adenine will pair with the thymine. Guanine will pair with the cytosine. In DNA. I am talking about DNA. Students, is this clear? Any doubt? Okay, yeah, this is very important. Now, so let me draw the next page. Just I'll show you this. So, adenine of one polynucleotide chain and thymine of another polynucleotide chain in DNA. I am talking about DNA, right? Between this, you'll get hydrogen bond. Actually, 2 hydrogen bonds are formed between adenine and thymine. Remember this. And between guanine and cytosine, okay. So I have guanine here and I have cytosine here. Between guanine and cytosine, we get 3 hydrogen bonds. So remember the number of hydrogen bonds between adenine and thymine, guanine and cytosine. Remember, adenine never combined with the cytosine. This combination is not possible. And similarly, guanine never combined with the thymine. Always adenine combined with thymine and guanine combined with the cytosine. Okay, so in these two chains, okay, if we have adenine, then this chain should have thymine. So the two chains are complementary. Students, observe carefully. They are not identical, they are complementary. What is meant by that? If I have adenine here, I have correspondingly thymine here. Okay, then guanine, cytosine, guanine, like that. Okay, so the two strands are complementary to each other. Students, is this clear? Any doubt? Any doubt, students? Yeah. right? So I have taught what is what are the sugar present in nucleic acid, what are the bases present in nucleic acid, okay, and what is phosphoric acid unit, and we discussed about nucleoside structure, nucleotide structure, and how one nucleotide join with the other nucleotide. Okay, and remember, yeah, here I like to take this point, students. Just note down this very important point. Under carbohydrate, very specifically polysaccharide, if you take a polysaccharide, in polysaccharide, one monosaccharide and another monosaccharide are joined together through ether linkage. This linkage, this ether linkage, is what we call as glycosidic linkage. Glycosidic linkage. Okay? Whereas in proteins are polynucleotides, polypeptides. One amino acid and another amino acid units. Okay, actually it's a chain. One amino acid and another amino acid are connected through a linkage given as CONH. CONH linkage is called peptide linkage. It is called peptide linkage. Students, very important for competitive examination. In nucleic acid, also called in nucleic acids, also called polynucleotides in nucleic acids. Okay, one nucleotide and another nucleotide, it's a chain actually, are connected through phosphate unit. O, P double bond O minus. They are joined together through phosphate unit, and this is called phosphodiester linkage. Students, remember this, very, very important. In polysaccharide, two monosaccharides are joined together by glycosidic linkage. In proteins, two amino acids are joined together by peptide linkage. CONH linkage is called peptide linkage. In nucleic acid, also called polynucleotides, in polynucleotide, two nucleotides are joined together through a phosphodiester linkage. Okay, students, remember, these are all very, very important points. Okay, now, then try to do the last point. Okay, in DNA, I have told you, DNA has two polynucleotide chains. Okay, so we have a double-stranded. Let me write like this: DNA, deoxy and ribonucleic acid. DNA contains, or DNA possesses, two nucleic acid chain, two polynucleotide chains. Okay, DNA contains, each DNA molecule contains two polynucleotide chains. Okay, and the two chains called, that's why it's called double-stranded. Okay, double-stranded. And the two chains, they wound around and form and exist as helix. Okay, so they exist in helical form, right? Alpha helical form, right? The two strands, double-stranded. Okay, the double strands, STRANDS. The double strands, they exist as alpha helical manner, alpha helical structure. Okay, not only that, the double strands are anti-parallel to each other. They are anti-parallel. You know very well what is meant by anti-parallel now. Okay, yeah. Students, just I will draw the structure. Just observe this carefully. Okay, this is a right-handed helix, right? Alpha helical means right-handed helix. Right-handed, okay. So I have drawn right-handed. Now let me draw another nucleic acid. This is one nucleotide chain, polynucleotide chain. I will draw another polynucleotide chain here. Goes like this. The two chains actually look like this. The dotted line indicates that part is behind the paper, and the thick line is in front of us. Okay, so it is nothing but right-handed helix. Okay, and students observe this, very important. This and this point. Okay. Okay, this is called one turn. I am taking adjacent similar points here. This is called one turn. One turn is called pitch. What is the pitch length? The pitch length is in DNA, the pitch length is 34 angstrom units. 34 angstrom units are 3.4 nanometers. Okay, this is what we call as the distance of one turn. Okay, one turn, right? Length is 3.4 nanometers in DNA, right? And if you see, in one turn there will be 10 polynucleotide, 10 nucleotides pairs will be there. Okay, so one turn, let me write, one turn contains 10 base pairs. 10 base pairs. Why I am saying base pair? Because if one chain has adenine, the other chain exactly has thymine here. So between adenine and thymine there is hydrogen bond. And here if I have guanine, this chain has cytosine. So between guanine and cytosine we have 3 hydrogen bonds. Okay, we call this as a pair, base pairs. Okay, in one turn, how many base pairs are there? There are 10 base pairs exist in one turn. Okay, so for 10 base pair, the length is 3.4 nanometers. Okay, then what is the distance between two base pairs? Suppose I have a base pair here. Assume that this is adjacent base pair. Let us assume adjacent base pair. What is the distance between two adjacent base pairs in DNA? For 10 base pairs, the length is 3.4 nanometer. So the distance between two adjacent base pairs. Okay, distance between two adjacent base pairs in DNA is equal to 3.4 divided by 10. I will get 0.34 nanometers. 0.34 nanometers. This is the distance between adjacent base pairs, okay, in DNA molecule. And see here it exists as helical, right? Helical structure. So there is a diameter here. What is the diameter? The diameter is 20 angstrom units. Diameter of any DNA, whatever DNA you take, the diameter will be roughly in the range of 20 angstrom units. Okay, but length can be any value. Okay, if you move from one DNA to another DNA, right, this length will differ. But these base pairs and the pitch length, these are all are fixed values for any DNA molecule. Okay, so keep this in mind, very important. Okay, yeah. Students, any doubt in this? Any doubt up to this? Okay, right, good. And these two is anti-parallel. What is anti-parallel? If one chain goes from 5' to 3', okay, if this chain is 5' to 3', the other chain will go from 5' to 3' in the opposite direction. Okay, so that's why it is called anti-parallel, right? And I have given what is the pitch length, okay, number of base pairs present in one turn, and distance between two adjacent pairs, right? So keep all these things in mind, and remember, one chain and the other chain, they are complementary to each other, because if one chain has adenine base, the other chain has thymine base, okay, they join together through hydrogen bonds. Okay, students, can you tell me how many hydrogen bonds present between guanine and cytosine base pair? Guanine and cytosine. 3, very good. How many nitrogen atoms present in adenine? Adenine. Purine, okay, purine base has 4 nitrogen atoms, I told you. But in adenine, extra amino group is there, right? So it contains 5 nitrogen atoms. Remember all the information. They may ask questions in any manner. Okay, so based on structure, they will ask questions, right, in any manner. So you have to be ready to answer those things, right? Yeah, students, any doubt up to this? Is it clear? Right. Okay, so with this we have completed nucleic acids and the structure of DNA, right? Now what I am going to do, what is meant by denaturation? Okay, denaturation. You know very well, whenever DNA molecules is exposed to a high temperature or exposed to some radiation, ultraviolet radiation very specifically, okay, or any reagent, if you add any reagent, what will happen? The high temperature or the radiations may break the hydrogen bonds between the base pairs. Between the base pairs, we have hydrogen bonds, no. So when ultraviolet radiation falls on that, it will break the hydrogen bond. What happens if it breaks the hydrogen bond? If it breaks the hydrogen bond, the two chains will start to move away. Therefore, the three-dimensional, see, I forgot to say this, I'm sorry. 3D structure. Students, 1 second. I'll be back. 1 second. Okay. So this is very important. The three-dimensional structure of DNA, okay, that is very, very important for the function. Right? You know very well the DNA has specific functions, okay. So the three-dimensional structure of DNA is responsible for its function. If high temperature or if radiation falls on the DNA and if it breaks the hydrogen bond, the two nucleotide chains, the polynucleotide chains will start to move away. Therefore, the three-dimensional structure gets disturbed. Right? In that case, the DNA molecule may completely lose the function. This is what we call as denaturation of DNA. Okay. And renaturation, that is the separated nucleotide chains may join and may get the original function, but this is happening in rare situation. Okay, in rare situation it may happen, but usually when the two polynucleotide chains starts to move away, that DNA will never function. Will never function. That's the point you should keep in mind. So three-dimensional structure is very important for DNA, for RNA, as well as for proteins. I have told you. Okay, this is very, very important. Right? Yeah. Then before completing this, students observe this carefully. Suppose I'll take a polynucleotide chain. It's a nucleic acid chain. Okay, and this is 5' end and this is 3' end. Always we see the bases from 5' end to 3' end. This is by convention. Okay, suppose I have here, I have say adenine, here I have guanine, here I have cytosine, here I have thymine. Like that I have bases, right? Cytosine, thymine, adenine. It may be of any order. Always we read the bases from 5' end to 3' end. Okay, the first three bases. See, adenine is present in one nucleotide. Guanine is present in the next nucleotide. Cytosine is present in next nucleotide. Okay, three adjacent nucleotide form what is called the codon. It is called a codon. Okay, and next three form another codon. Okay, so we have codon structures. These codon structures are responsible, okay, for the synthesis of proteins, right, in human and animal bodies. Okay, we are not going to discuss that here. Okay, you may learn all these things in your higher classes. Okay, right, students, I stop here. We'll continue. Second.